Bismillahirrahmanirrahim. This is Biochemistry 2 course, code Chem 342, and today we are having lecture 29. Our today's topic is vitamins A. This is Dr. Naeem Khan, Chemistry Department, CUS. Need to have a start. Outlines of today's topic. We will discuss about the chemistry of vitamin A, then about the metabolism of vitamin A, how vitamin A is taken in the food, how it is changed, and how it is absorbed. We will talk about the different sources from animal and possibly from plants of vitamin A available to human beings. And we will talk about the human requirements of vitamin A, how much amount of vitamin A is required to humans. Uh, I mean the recommended dietary allowance. And we will highlight the important functions known for vitamins A. Need to have a start. Chemistry of vitamin A. Vitamin A is known as anti-xerophthalmic factor, or it is also called bright eyes vitamin. Why? Because of its established role in the visual process and because of its established role in the skin maintenance. Xerophthalmic. So its vitamin A is called vitamin bright eyes vitamin because it has a very much established role in the eye vision, and also it's a very important role in the skin maintenance. So it is also called anti-xerophthalmic factor. Vitamin A basically occurs in two forms, and they are designated by vitamin A1 and vitamin A2. And carotenoids are actually known as provitamins of vitamin A. Means carotenoids are the forms from which vitamin A are produced, and basically two carotenoids, alpha carotenoid, no, three carotenoids, alpha carotenoids, beta carotenoid, and gamma carotenoids, plus cryptoxanthin. They are the carotenoids which are the provitamins of vitamins A, means they are the forms from which vitamin A are produced. Beta carotenoid is basically the one which is the most potent carotene of all, and this is the one usually taken in the raw form as provitamin in the food. Beta carotenoid structure, if we discuss, it is made up of 8 5-carbon isoprenoid units, totally making 40 carbon chain, and the 40 carbon chain is having anion ring at each end. This is the structure. Here we have 40 carbon chain is a 5 5-carbon isoprenoid units, 8 units, and on each end we have beta anion ring. Beta anion ring. This is the point where this chain will be broken and we will get vitamin A. So beta carotene is the most common provitamin of vitamin A that is taken in the food, and its structure is containing 5-carbon isoprenoid units, 8 in number. They are held together with beta anion ring at each end. On hydrolysis, 2 moles of vitamin A are produced. This is the hydrolysis. 2 molecules of water are added. Here the bond is broken and we get 2 moles of vitamin A, which is an alcohol derivative. So this bond is broken and we get 2 vitamin A molecules from 1 beta carotene. So vitamin A, basically vitamin A1 and vitamin A2 There are carotenoids, alpha, beta, gamma, or cryptoxanthin, which are the provitamin forms of vitamin A. Beta carotene is the most potent form, and beta carotene is basically having five carbon isoprenoid units, eight in number. They are held together with beta ion rings at the two ends. On hydrolysis, the most common beta carotene, which is taken in the food, is broken down, and we get two molecules of vitamin A. This can be traced in the GLJ fundamental biochemistry. Chemistry of vitamin A continued. Vitamin A2, which is the other vitamin, we saw in the last lecture, vitamin A. Vitamin A2 is the other vitamin, and this differs from vitamin A. How? Because it possesses an additional conjugate double bond between carbon atom of 3 and 4 of the beta ion rings. Here we can see the structure. This is carbon number 3 and carbon number 4 of the beta ion ring. Here, this bond was saturated in the A1, but in A2, it is having this double bond. Because of this double bond, vitamin A2 is different from vitamin A1. Its potency of vitamin A2 is 40% that of A1. A1 is more potent, but A2 is 40% of A1. This structure can also be traced in the fundamentals biochemistry by GLJ. Metabolism of vitamin A. Here we can see different conversion of beta carotene takes place. Dietary beta carotene, which is taken in the food, it's split into retinol by an enzyme of the intestine. This is a beta carotene, and it is broken into the aldehydic form retinol by enzyme of the intestine. This retinol is then by reduction is converted to retinol. If it is reduced here, then from retinol we get retinol. Retinol is converted to retinol ester by reacting to a fatty acid. Then this retinol, by reacting to any fatty acid, this can be converted to retinol ester. This is a more stable form, and usually it circulates in this form. Retinol ester is enzymatically hydrolyzed to retinol, which is esterified. This can be by hydrolysis be converted to retinol, and this retinol is actually the active form. Whenever required, it will be converted to retinol. Otherwise, it will be re-esterified, and the stable form can exist. This is absorbed through lymphatic system and stored in the liver. Lymphatic system absorb this esterified retinol, and this is stored in the liver. Beta carotene is basically the raw provitamin form of vitamin A, and it is split by the enzyme, intestinal enzyme, into retinol and aldehyde form. Look here, the aldehyde bond. Then it is reduced to retinol, which is the active form of the vitamin. But whenever this vitamin is not required, this is esterified by forming ester linkage to a fatty acid and absorbed and is transported to the liver where it is used. This figure can be traced in the G.L. Jain book, Fundamental of Biochemistry. Metabolism of vitamin A. Liver stores vitamin in large quantities, mainly in the form of retinal ester. Yes, as we saw in the last slide, that retinol after retinol is required in the active form, but it is esterified with the ester, and the esterified form is the stable form, and it is stored in this form in the liver. Vitamin A circulates in the blood as retinol bound to retinol binding protein. It is actually retinol is the vitamin A is fat soluble vitamin, so it cannot freely move in the blood directly. because blood is aqueous. Usually it is circulating in the blood but is bound to the retinol binding protein. Retinol ester in the liver has to be converted to retinol before it mixes with the blood because the we saw that by hydrolysis the retinol ester can be converted to retinol, and basically vitamin A is the retinol. So whenever it is circulating in the blood, we say that it is bound to the retinol binding protein. So it is actually hydrolyzed to the retinol form, and then it can pass to the blood. Liver can successfully convert to retinol to retinol and retinol or to retinoic acid. In the last slide, we saw that retinol can be converted to the retinol, base converted, or retinol can be converted to the retinol. This is done by hydrolysis or reduction, and this can be converted to retic acid, whether oxidation. So these are the three forms interconvertible within the liver, and these reactions are possible. Retinoic acid is absorbed from the intestine and is rapidly excreted back into the intestine through the bile. The retinol can pass back to the intestine through the bile, and here it can be absorbed from the intestine. This retinoic acid is most suitable for absorption from the intestine. We saw that liver stores vitamin A in the retinol ester form. Whenever retinol forms ester with a fatty acid, it is called retinol ester. So this is the form, uh, storage form of vitamin A. And vitamin A is fat soluble vitamin, so it cannot circulate freely. It can circulate in the blood bound to the protein. There is retinol binding protein RBP, and retinol ester in the liver has to be converted to retinol because retinol is the vitamin A form, active form, and if, before it combine to the retinol binding protein, it must be converted to retinol, and then it will maybe pass to the blood. Liver can convert the conversion of retinol to retinol and retinol to retinoic acid. And important is retinoic acid is the form which is absorbed from the intestine, and also it can be passed back to the intestine through blood, through bile. Coming to the sources of vitamin A sources, if we look into the beta carotene vitamin in the fruits and vegetables, here we can see that the important fruits in which vitamin A is possible are mango, which is having up to 300 microgram per 100 gram of fruit. The similar with orange, we have 200 microgram per 100 gram. And similarly, tomato is having 190, but the highest ratio is the here drumstick leaves, 7,000 to 8,000, and coriander leaves, that is up to 7,000 microgram per 100 gram. Similarly, carrot is having very high concentration, and that is 1,500 to 3,000 microgram per 100 gram. Radish leaves 3,600, and mint leaves is also having 1,800, and cabbage is having 1,300 microgram per 100 gram. So the vitamin A rich sources are the drumstick leaves, coriander leaves, which are having in thousands, 7,000 to 8,000 microgram per 100 gram of sample. Similarly, the next highest source is the radish leaves, which is 3,600 microgram per 100 gram. These are the common food we take daily basis. So we can say that the plant world, Vitamin A is widely distributed and its sources include mango, orange, tomato, drumstick leaves, coriander leaves, carrot leaves, radish leaves, mint leaves, and cabbage. And it's similarly, and the animal foods. From the animal food, these have different sources, and where we can get this vitamin A as in our food. Here we can see that the cod liver oil, these are actually the using as a source of for the commonly recommended as sources of vitamin A, cod liver oil, or these are actually fish liver oil. Actually, these fat vitamins are oily, soluble in fat. So usually the liver oils are the sources of the fish liver oil. So this cod liver oil and this is used as the recommended source of vitamin A and is containing 600 international unit per gram of the food. One international unit we will discuss in our later coming slides. Similarly, other sources are also available here. Southern elephant seal, enzymatic Hensky, yeah. Similarly, other foods are available, but here these are the international food. For our food, the recommended is the cod liver oil, which are contain a high concentration, and the recommended is up to 600. Similarly, some foods we are having 12,000 to 14,000 or 24,000 to 35,000 international units per gram of the food. These source tables from the animal and plant can be traced in the GLJN biochemistry book, Fundamentals of Biochemistry. Requirements of vitamin A. The recommended dietary allowance RDA of vitamin A is about 5,000 international units. Daily we need 5,000 international units of vitamin A. What is the international unit? According to the World Health Organization, one international unit is the biological activity of the 0.000344 milligram, which is equivalent to 0.344 microgram of the synthetic vitamin A acetate, which is equivalent to 0.30 microgram of retinol. How much we need? Daily dietary allowance is 5,000 international unit, and one international unit is 0.30 microgram retinol. So for our normal health, it is 0.30 microgram retinol we need for our as a 1 unit, and we need up to 5,000 in our diet. Less toxic if taken more vitamin A. Vitamin A overdosage is not that much dangerous. However, if we take more than 50,000 international unit of vitamin A, then it will cause swelling of bones. Coming to the functions of vitamin A. Vitamin A is essential for health, cell growth, immune function, fetal development, and vision. In general, vitamin A is having so many functions, and that's required for good health, cell growth, immune function. Immune system is depending upon vitamin A2 and the fetal development. But the prime function of vitamin A, which is vitamin A, is more known for the vision, eyesight. One of the best known function of vitamin A is its role in the eye health. For the vision and eye health, this is a very much important function of vitamin A. Actually, retinol, which is the active form of vitamin A, combined with the protein opsin in the eye, there is a protein opsin, and when this retinol combines with the opsin, it forms rhodopsin. And these are the molecules which are requirement for color vision and low light vision. Here we can see this is opsin molecule, and when it combines to the retinol, vitamin A, we get rhodopsin. Actually, these are the rods. This rhodopsin is very important for the low light vision or vision in the dark, or color vision. So it make available. But this compound is sensitive to light. Whenever someone switch on bulb light, this compound decomposes and we get opsin and vitamin A. And then a person, this we can see that whenever light goes and in the dark, in the first few seconds we are unable to see anything, but after some time we gradually becomes able and our vision to some extent restore. And this is because in that time the option combined to vitamin A and rhodopsin is formed, and this rhodopsin enables us to see things in the dim light. So the most important function of vitamin A is it's combined to opsin to form rhodopsin, and this rhodopsin enables us to see things in the dim light. This picture can be traced in the GLG and biochemistry. Another function is the vitamin A is it protect and maintain the cornea, eye cornea, and we know that cornea is the outermost layer of eye, and the conjunctiva, which is the thin membrane that cover the surface of eye and inside of eyelids. So it's maintaining the health of eye because cornea and conjunctiva, these are the two things which are requiring vitamin A for its normal health. Vitamin A helps maintain surface tissues of the skin, intestine, lungs, bladder, and inner ear. And that's because of its role in the skin. It is known as integer of thalamic factor. So this is requiring and for the normal health of skin too. Also the lining of intestine, lungs, bladder, and we know that these are all important tissues. Maintaining of the surface lining is very important, and vitamin A is required for that. Vitamin A supports immune function by supporting the growth and distribution of T cells, a type of white blood cell that protects our body. T cells have different forms, and actually in the immune system we have basically two mechanisms. One is through uh, T cells, and the other is to the antibodies. So this is one of the mechanism which is required by the body to control the antigen. So T cells growth and distribution is dependent upon vitamin A, and thus vitamin A is required for our immune system. Western Todd book is recommended, and GL Jain book, especially GL Jain book. Please, students, thoroughly elaborate all the points we discussed today. From today, uh, I will say goodbye. Thank you. See you next time. Allah Hafiz.